While we're at sea, the students broke up into 4 main groups. I was fortunate enough today to follow along with the visualization group. What is this group doing? Well, they're actually trying to visualize marine microbes, the stuff that you can't see with the naked eye. I mean, it's amazing. This water is so blue, yet it's teeming with life. They are using microscopic techniques, looking under a microscope to see this material. They're also using a flow cytometer, which allows you to count this stuff. We are very fortunate to have Dr. Mike Siraki from the Bigelow Laboratory for Ocean Science on the cruise with us, leading the microscopy lab.
So we're preparing samples here for microscopy to look at bacteria, and we're staining the samples with DAPI. And DAPI is a stain that lights up the DNA in cells and allows us to visualize them under the microscope.
My name is Charles Schutte. I'm a second-year PhD student in the Department of Marine Sciences at the University of Georgia in Athens, Georgia. This is Mike Siraki's, flow cam, which basically shoots a laser through a stream of sample that comes in through the instrument that we collected from the ocean. And then anytime that laser hits certain types of organisms that have pigments that they use to harvest energy from sunlight, those pigments fluoresce and release a different wavelength of energy from what they're being hit with by the laser. And when the computer gets that signal, it sends a signal back to the instrument that says, take a picture now. And so what we get is a series of pictures of the different microorganisms living in the water.
My name is Maria Hamisi from the University of Dodoma in Tanzania. I'm working there as a lecturer by the time being, but I'm here as a student of the CMOR course. And I'm in the visualization part, which we are collecting samples to view in the microscope, flow cytometry, like counting the things that we cannot see with the microscope.
Flow cytometry is a technique used to count single cells. So we're actually running ocean samples through our instrument back here, and it's quantifying the different bacteria that we see, including photosynthetic bacteria like Prochlorococcus, Syndicococcus, and heterotrophic bacteria that have no pigment. Well, so the primary cell that we look at out here is Prochlorococcus. And Prochlorococcus is the most populous phototroph on the planet. A phototroph is a cell that contains chlorophyll or some other pigment that performs photosynthesis.
Please follow along during the next 3 days as we will visit with the other groups of students.